Hi, everyone. My name is Wale Salahuddin. Today, I'll be presenting some work on a method we've been working on to address sort of problems where we essentially have some mechanisms that we would like to measure from data. However, we don't necessarily know sort of the details of the mechanisms in the data. So today, I'll be presenting separating neuroencoding and decoding pathways in fMRI. By disentangling causal and anti-causal mechanisms. And my ABM advisors are Samni Koichal and Brad Sutton. So some notation or terminology before I start. So we'll define two type of mechanisms, causal and anti-causal. So these will be defined with respect to an outcome Y, and we'll call them causal mechanisms, mechanisms that cause Y, and then anti-causal will sort of call them the effect of Y. And so these two terminologies will be used throughout the rest of the presentation. I want to talk about causality and anti-causality in fMRI, or really generically, you can think of just any sort of process you're interested in where you observe pre-outcome measures and post-outcome measures. And so here we typically observe an fMRI scan, and then we observe some behavior or experimental state. And our goal is we want to be able to reliably predict and understand mechanisms for the behavior. And so we want to understand sort of the encoding mechanisms that are upstream of the behavior, and then we want to understand decoding mechanisms that are downstream of the behavior. And so these are causal and anti-causal respectively. So the challenge here and in many other problems is resolution. So here for fMRI, it's temporal resolution. And so for instance, we might observe both the encoding and decoding pathways simultaneously just because the fMRI isn't as fast as the processes we're interested in capturing. So for instance, in this figure here, we might have some behavior that's sort of being, that's occurring at these purple time points. And then we might have the encoding mechanisms that are occurring as well, the decoding mechanisms that are occurring as well. But here we're actually taking a slice or taking measurement at like these time points right here. And so we see mechanism overlap. So for like a specific slice, we might observe activations that are both because of the encoding pathways and the decoding pathways. And it becomes, if we want to understand sort of what those pathways are, it becomes important to learn to disentangle these two things. We can talk about this general model that I've been working on, which tries to disentangle these two mechanisms. So we can look at the graph here on the right. So we can sort of think about this as the generative process. So this is our assumed way that the process occurs. So here we have some ZC, which we'll call our encoding features, which causes our behavior or has a path to our behavior. And then we have some post-behavior measurement as well, which we'll call our decoding features. And then what happens here is actually we don't necessarily observe these two separately. We observe some function of them that comes together in our X. This might be our fMRI scan or our observed features. And we also observe the behavior. And then we also assume that we have some like latent factor that affects everything. So this might be machine setting, this might be individual differences. This might be sort of something that's maybe unobserved, but we have some E, which we'll assume is sort of like a sufficient statistical view that can tell us what these differences are. So this might be just like subject label. This might be the site where the measurement was taking. Information like that could be considered our E. And so what we would like to figure out is what the encoding pathways is. So we want to figure out this mapping from ZC to Y. And we want to figure out the decoding pathways, which is our mapping from Y to ZAC. And this can allow us to sort of understand what the two different mechanisms are. So again, the question is now, how do we disentangle these two processes? Because recall, we only observe this X, which is a function of the two feature sets, and then our Y, our behavior. And so here's our learning setting for our method is essentially, we're going to expect to see multiple different measurements. And so we want sort of these things called domains, which is sort of like distinct examples. So this could be distinct individuals, this could be distinct machine sites, or et cetera, just something that's like sort of different in how the data was acquired. And then we're going to assume that we observe A, the fMRI scan, and then B, the experimental state or behavioral data that we actually are interested in learning mechanisms that are related to. So the goal is we want to learn mechanisms that are shared across domains. And so shared across domain, meaning that it's robust to various things that we don't care about. So like it could be robust to individuals. So that means these are sort of like shared mechanisms that occur in all of our examples, or it could be robust like machine settings. So again, we may not want to capture changes that are induced just by the machine settings that may vary from study to study, trial to trial, individual to individual. And so we go back more generically to our assumed generative mechanism. So again, we assume that our data is generated by this sort of causal graph, this structure. And a few things we can read off the graph, making some like assumptions about DAG properties. We can see that, first of all, our Y, our predicted, the behavior that we're actually interested in understanding, we can actually make it independent of the unobserved confound if we condition on the causal features. And so this is a property of just like causal features in general, is that they can sort of separate, condition on them, we can separate the behavior that we're interested in learning from like the spurious information that we don't think actually matters to the understanding of the process. And then second, the other thing is that if we look at our encoding and decoding features, essentially they're also conditionally independent given our labeled and then our unobserved U here. But again, since we assume that our E is sufficient statistics for this U, we can observe this sort of E and then be able to say that it's also conditionally independent given Y and E. And so these two properties motivate our method and gives us a way to sort of try to disentangle these two mechanisms. And so our learning strategy is going to be sort of this long-term process where we try to go from our fMRI scan or our observed features, and we'll reconstruct it into whatever feature space we feel is most appropriate. So this could be just like brain region modes. And so we start from this X, and what we do is we learn two feature extractors to try to map to our causal features and our anti-causal features, or our encoding features to our decoding features. So we learned these two models. So this could be your favorite featurizer. It could just be like a linear model. It could be a deep learning model, whatever makes sense for your problem. We basically implement two of these, and then we implement two predictors. So these will represent our actual mechanisms, our encoding and decoding mechanisms. So here, our causal predictor will be mapping from the output of our causal feature extractor, our causal features, to our causal prediction, and then we'll be mapping our anti-causal features through this full predictor to our correlational prediction. So again, recall that we want to learn mechanisms that are sort of invariant and robust to various things. This will be our prediction or the explanation of our process that's robust to those spurious things. And this would be sort of like the generic, what's the best we could do if we didn't care about what's robust and what's not robust, to just predict and understand our features. And so this is here just for technical reasons that we need to predict this outcome as well. And so for the sake of completeness, I have the slide here, but I'm actually going to skip it and just mention that we essentially implement this by basically adding a regularization term to our loss function in our learning model to match each of the properties of the DAC that I described before. And so the takeaway is that many problems follow this causal and anti-causal formulation, some of them including really a lot of things that are sort of biological, where we don't think that our measurement devices are per resolution of the actual biological processes going on. Our proposed method sort of disentangled pre-behavior and post-behavior features. So we can sort of disentangle these two processes to give us a better understanding of the mechanisms going on and what features are actually encoding versus decoding the outcome of interest. And then the third point is that in addition to many other applications, this is specifically relevant to fMRI, where we know that the temporal resolution is not quite at the speed, and we're constantly trying to sample faster. But this might give us a way, when we can't sample faster, to still be able to glean some information from the observed data. And so the next steps is we've sort of applied this method because it's so generic in other domains. The next task is to sort of find the appropriate task fMRI data, figure out to evaluate this method on, figure out the appropriate feature spaces, the right metric of success, because we don't necessarily have ground truth of what encoding and decoding pathways is in a lot of applications that might be interesting. And then also the right set of domains and what the implications are on the sort of generalization and the information we glean. So this could be across individual differences versus machine-induced differences. And with that, I'd like to acknowledge my advisors and the MBM program, and I'll be happy to take any questions now. Thank you all for listening.